Yeah.
Hello, everyone, and welcome to one more event of Pint and L Taos. My name is Laura Luzia, and I'm the city coordinator of Pint of Science Amsterdam. And with me is Taishi Oshi. He is the city coordinator of Pint of Science Netherlands. And tonight we will be talking about virus and SARS-CoV-2 that causes the disease COVID-19. And our guests are Ellen Carbo and Xavier Martinez. I hope I'm saying it well. Both are PhD students at the Leiden University Medical Center. Hello, Ellen.
Hello.
Hello, Xavier.
Hello.
Welcome to tonight's event.
And
Ellen
don't
and
forget to toast to our new guests.
oh no, of course no.
So
Prost.
cheers, guys,
Cheers.
for coming along.
Cheers.
Thanks.
So, yes, Ellen and Xavier will be talking about virus. How do they cause infections and how can we detect them? So this is a very hot topic at the moment, not for the best reasons, and we really hope that soon we can get back to our normal lives. So please feel free to ask any questions. We are streaming via YouTube and Facebook, so you can just type your question in the chat. And yes, I think that's about it. The floor is yours.
So I guess I'll be starting. As Laura said, I'm Xavier. I come from Barcelona, and I'm in my second year of PhD here in Leiden at LUMC. And I started working with coronaviruses before the outbreak of SARS-CoV-2. And yeah, I'll try to explain a little bit the basics of a virus infection, coronavirus infection, and then Ellen, my colleague, will take the floor. So, as Laura said, SARS-CoV-2 is the responsible virus for the disease COVID-19, and SARS-CoV-2 stands for severe acute respiratory syndrome, coronavirus 2. And why the 2? That's because already in 2002, 2003, there was an outbreak of a very similar virus, and by that time it was named SARS-CoV, SARS coronavirus. And well, as we all know, the first outbreak was identified in China about 10 months ago, and by now it has spread to every corner of the world. And at the moment, there's almost 40 million cases reported, and a bit more than a million dead people because of the virus, although these numbers could be even higher. And well, SARS-CoV-2 is is an airborne virus transmitted via the air, and that means that if we are in a room with someone who is infected, that person will exhale or will sneeze, cough, and that will generate droplets. These droplets may contain the virus, and then we'll end up in different surfaces that we can touch with our hands, and then at some point touch our face with the hands. And at that point, the virus can make it to the respiratory tract. But then what happens? So viruses, all of them are obligatory intracellular parasites, meaning that in order to multiply, they need to go to get into a cell. And how does the coronavirus do it? So as we have seen the cartoons or schematic representations all over the place, the coronavirus looks like that. It has some spikes protruding from the membrane, and these spikes, the virus uses them as if they were keys to unlock a door in a way. So these spikes will bind to proteins in the outer membrane of a cell, and after binding, it will start making it into the cell. So actually, most of the vaccine strategies directed against the viruses aim to develop antibodies in our body that would block these keys from making contact with the proteins in the membrane of the cell. But if that's not the case, so far we are not at that point. point yet. The virus makes it into the cell, and then what happens? Well, the first thing the virus tries to do is not to be noticed by the cell. The cell has many mechanisms to police against intruding agents, and that includes viruses. So the viruses will try to counteract that policing mechanisms with its own strategies. So first of all, when the virus goes in, the genetic material of the virus will be exposed in the cell, but this genetic material is dressed up to look as if it was cellular, or in this case, a human genetic material. And then this genetic material will be converted into proteins by machinery of the cell. So the virus will make use of the machinery of the cell to translate, to convert its genetic material into a protein. For coronaviruses, the first protein that's being generated in the cell is a very big protein that is not 100% functional. And this protein needs to be cleaved in smaller proteins in order for each one to perform its function. I like to compare it to a roll of garbage bags. At the end, maybe you can use it for something, the whole roll, but you need to tear apart each bag for it to have its purpose, its function. So yeah, how does this cleavage of this big protein happens? So within the big protein, there's two proteases. Proteases are proteins that are able to chop other proteins. So these two proteases will cleave this big protein into 16 non-structural proteins. And by non-structural, I mean that they will not end up being part of the structure of the virus. Rather, they will be building blocks of a factory within the cell to generate new virus particles. So all these individual proteins will have certain roles in order to generate what will become later the progeny viral particles. And
sorry, Xavier, to interrupt quickly. It sounds like we know quite a lot about the SARS-CoV-2, how coronaviruses work. So what's the difference right now? Why can't we apply the same strategies? Is it we as a human host can't identify it earlier, or is it that there's certain mechanisms that make this specific strain of coronavirus different and make it hard to interact? Hard to defend against, I guess.
Well, I don't really fully understand what you're asking.
As you mentioned that we understand how coronavirus works. Sorry, I guess viral genomics is not my strength. So it's more along the lines of breaking it down simply. You said it's sort of like two garbage bags and then breaking it down. That's I guess, putting it very simply. What's the new novel area? Is it like one garbage is stinkier currently, or just harder to break down, that's causing so much of the rapid transmission? Why is it sort of expanding so quickly? That's from a few cases last year, now it's making us all work from home.
Yeah, well, I wouldn't say it has to do particularly with this big polyprotein and the cleavage of it. Yeah. There might be other factors involved. Mm-hmm. So, there's been this suggestion that in the spike protein, when I was explaining the viral entrance, there's a specific cleavage site for a host protease, and this host protease is present in many tissues in the body. Whether for the other first SARS coronavirus, this cleavage site in the spike protein was not there, and that might play a role in the tropism of the virus, as well as to the egress or the release of the virus from the cells. But and then of course there's different things like, this virus, it is fairly pathogenic, but it's not like an Ebola, So there's many asymptomatic cases or people showing up with very mild symptoms, and that makes it easier for the spread of the disease, because the people will not notice that they are very sick, or they will not notice soon enough. So that also
Exactly.
plays a role in the spread of the disease.
Okay.
I actually had one question. Well, maybe both of you can try to answer. So, what really, yeah, makes me think about the virus is how can it be that there are so many people without symptoms? Because, well, I guess this is one of the strategies of the virus to being propagated so easily, because people don't know that they are infected, so they just keep infecting other people. And yes, I was just wondering how the virus well escape the immune system in one way, and in another way, if there's any idea now about the percentage of people that don't show symptoms. And why does that happen? Why do some people develop symptoms and others don't?
Should you answer that, Ellen, or should I go for it?
Well, I think you're the expert on this part of the virus.
Well, not really. I'm very in the molecular part, but yeah. Well, I think, as it's been said all over the news, it really depends in order to present symptomatology on your age, even your gender, your previous diseases. So your basal innate and immune system will partly determine how the disease will develop. And then, of course, the virus has strategies to try to corrupt the immune system. I was talking about the proteases. So the proteases are known to have accessory functions. As I said, they cleave the big protein, but they also cleave other proteins inside the cells. And part of these accessory functions, this cleavage of several proteins, are attributed to corrupting the innate immune system, or to blocking it from responding to the virus.
Okay, so that means if there are two people in the same room and one person has, let's say, uh, his or her immune system, it's a little bit lower, or it's not functioning properly, that person has a higher chance of getting the virus if both of them are exposed to the virus.
I would say the chances of getting it are the same. It's just the chances for the virus to replicate and spread around the body are different then. So the chances
Okay.
to get it, but then the person who has, let's say, a better immune system won't develop the disease to such an extreme. It will
Okay,
kind of
so
uh,
both of them will be exposed and they will develop antibodies, I guess, but one of them will develop symptoms and the other will not develop symptoms.
yeah, that's a big idea. And then, of course, depending on how long you have it, what's the viral load you have it, and the number of antibodies you develop will be different. But yeah, that's pretty
Okay.
much yeah.
Okay, maybe just one more question, because yeah, I think this is really fascinating. So I was wondering if there is any study where people have taken some kind of medication to improve their immune system, and if these have shown an effect on how coronavirus can affect you and your health.
Yeah, I think so. There's been, yeah, from what I know, so different strategies. So one strategy is to give interferon. Interferon is a protein, well, to a component of the innate immune response. But I'm not sure whether the results of implementing that treatment are very conclusive. And then the other thing is try to put the immune response down, because at the beginning the virus will try to suppress it, but later on, normally the people who end up having severe problems are those who develop a too strong immune response, and then that's when the cytokine storm, this phenomenon, takes place, and then the other immune treatment is try to put down this response.
I see, but
it fairly works.
Do you think that I don't know, so my question was more in the sense of a prophylactic situation. So, you know, I know that well, now the virus is around us. Should I take some kind of medication to boost my immune system? Has every I don't know, any group look at it? Does this make sense? I don't know. I have no idea.
I have no clue either. There have been studies also started about tuberculosis vaccine, that people that are like a year ahead vaccinated, that maybe they have then have a better immune response later on. But I have not heard about any studies like if you eat more oranges, that then you're better off. I think
getting the food. Exactly.
Yeah.
Okay. Actually, sorry, Xavier. I think we've sort of bombarded you with questions. But as I guess as a key takeaway, what would you say again is for us to define regarding the SARS-CoV-2, its difference in the viral life cycle. So the key takeaway is viral protease is the main differentiator, or the one that makes a difference in how it's currently affecting us, and also how it's also affecting not just transmission rate, but also how it's affecting the illness.
No, I wouldn't attribute it to proteases.
Uh-huh.
No. I was just now going into proteases because that's the topic I work on. But the message is that, in my opinion, we still are getting to know the virus. Of course, in 10 months, we've done a lot. The scientific community has done a lot, but that we got to be patient in a way, and little by little try to understand it. We've been kind of notified before in 2002, 2013 that this can happen, and now it happened, and it happened at a bigger scale. And also another take-home message would be that we really need preparedness, because now, of course, we all rush trying to find something, but prior to that, we need to be prepared. And I would say for the future, we need to be prepared, and I think that pretty much links to what Ellen will talk about. So I would say, I wrap up here, and Ellen will link to that, I would say. Yeah.
Well, thank you, Xavier. So this is my colleague, Xavier, and he works in our research department. I work for the diagnostic department of the medical microbiology at the LUMC. And this is the department where we diagnose patients, and we do so by identifying microorganisms and pathogens. And there has been a lot about it in the news because, of course, we do a lot of, we try to identify a lot of bacteria, for instance, but we also do viral testing. And one of the tests we're really busy with is the qPCR of detecting SARS-CoV-2. And I will quickly explain you some things, how this works, because it's a very hot topic. And a lot of people, they say it's too sensitive. Other people say it's a fake test, but I will just take you through it. It's just a small few steps. So, Ty, could you maybe show the slide or touch it? One further. Yeah. So this is basically the qPCR in the very simple steps. So maybe a few of you are already been tested recently for SARS-CoV-2. So people are getting swabbed, and once this swab material is delivered at the lab, the first thing we do is that we extract the genetic information. So we extract the nucleotides, which consist of RNA and DNA. And then RNA is not very stable outside of the body. So the first step we do is that we actually copy the RNA into DNA. So then we have something we call cDNA or copy DNA. And then actually the real test starts, which we call qPCR or real-time PCR. And this is based on fluorescent signal and based on the amplification of the material that you have. For this, we need specific primers for a virus. So we have actually primers that were developed for SARS-CoV, for the E gene, but it also works because it has the same nucleotide sequence as SARS-CoV-2. It also works for the SARS-CoV-2 virus. And as there's no SARS-CoV anymore, we now use it for all the patients that show up recently. So the primers, they attach to the DNA. And then when the primer is attached, we can add an enzyme that builds in new nucleotides to make a copy of the first strand. Doing so, there can be a fluorescent signal either exposed by something that's called cybergreen or something that's called TACMAN. After this new strand is there, we increase the temperature of the PCR machine, and then the strands, the first strand we had and the copied strand, denature. So then we have two strands again, and then the process is repeated again and again and again. This goes on for 40 cycles. And at a certain amount, the amount of fluorescent signal exceeds a threshold that we can actually detect it. And because we amplify the material in every step, it works that if you start with already a lot of viral particles in the beginning, the sooner the fluorescent signal will also pop up. And then, I don't know if it's very good visible in the slide, but the orange signal, that only needs a low number of cycles before the signal pops up. Yes, that's over there. And then the blue one, that takes a bit longer. So that's a person with less viral particles. Then we also have the purple, it's again lower. And if you would have no viral particles for SARS-CoV-2, then this test would also would not show any fluorescent signal, and there would just be a flat line. And this is actually the way how we test if people are positive or negative for SARS-CoV-2. People say the test is not fast enough, but actually the test is only a few hours, but it's more the logistics around it, why people sometimes have to wait for days for the test results. But I think you could do this entire test within a few hours until you have the results. The only downside for this test, we all test patients for SARS-CoV-2 because we expect it, but we also test in our laboratory for other diseases and other viruses. But then you always need to know which virus you actually are expecting. So for instance, we also have diseases like encephalitis, and then we test 6 viruses that are mainly causing this. But if these people stay negative for all these 6 viruses, then for us it's still a puzzle actually how to diagnose a certain patient. So what we do then is we have a test as an add-on test, which actually called viral metagenomics. And that's all where my PhD project is also about. And metagenomics means that you look at all the species in a given sample, and you do so by looking at all the genetic material in a sample. And then I specifically look at the viral material. And then actually we can look at all the viruses out there. So we are not really in need of a certain primer set, but we just look at all the viruses at once. But also this works with databases that only contain viruses that we already know. So there's thousands of viruses and thousands of genomes that we already know. But now recently we see there's still emerging viruses, and at the LUMC we actually wanted to be ready. So what happens if next time there's no emerging virus in China, but what happens if it emerges in the Netherlands and it comes to our hospital? We actually want to be able to detect and identify this novel virus as quickly as possible. So that's why we wanted to validate the virus discovery model with this metagenomic test. Therefore, I took a few samples to start with. Maybe we can show the slide.
Mm-hmm. Yeah.
Yeah. So here we've the slide with the samples that I used to validate my test. So in order to actually see if we could use this test for virus discovery, we took a few different coronavirus samples. We had some patients that were infected by SARS-CoV-2. Unfortunately, or maybe better to say fortunately, we did not have any patients with MERS or with the previous version of SARS. Therefore, I took patients that were negative for any virus in their respiratory material, and then I added the cultured mixture of MERS and SARS to it, but also in a way that the amount of viral particles that I added was also a bit equivalent to an active infection. And then I actually I have to actually start to classify this. So I use a test which is called next generation sequencing. Then you get long strings of the DNA code. And these long strings of DNA code, I actually try to, by data analysis, I try to see where they matches genomes of already known viruses. Only now I had to make databases from previous years to actually see how this would work if you would discover MERS in 2012 without knowing you had actually MERS in your sample, if you could see to which family it would actually go. And then if we can go to the next slide, because I use the software tool called Centrifuge, and then we get actually like circle graphs that you can see on your screen. And in the top left corner, for instance, were the results of my SARS-CoV-2 samples. And then actually most of the samples are classified as SARS coronavirus, so the previous ones, because this was a database that I used from December 2019. And for instance, in the upper right corner, we have the SARS-CoV sample. And therefore, I actually made a database that only contained viruses from before November 2002, when the index patient was identified. And then we see it was, for instance, also classified as bovine coronavirus, because that time we were mainly aware there were coronaviruses infecting animals and not humans. So these were already sequenced, and the genome was known yet, but not too many bat viruses or other human coronaviruses. So these results already indicate to me that in my results I have a coronavirus. This program also would show me the amount of reads that are classified this way, but it still does not give me information of how much of the nucleotides actually resemble the coronavirus that I find. And I think that's useful information too, because you need a certain percentage to say if it would be closely related, yes or no. So I also tried different bioinformatic methods. I use a lot of bioinformatics for my data analysis, and that's actually that you just use informatics for your biological problems. And I used a method called the novo assembly, and I can actually explain this. So say if I would have like sequence read, so if this would resemble a string of just DNA, so I just get strings of 150 letters, nothing more, just 150 letters that contain the genetic information. The only thing, I would have like more than 10 million of these. And then my programs will actually start to build a genome from scratch. And they do so because they're going to check if some of the reads have genetic codes that resemble each other. So if all from here to here, the DNA code would be the same and it overlaps, then it would start to build a genome part. So it goes on and on and on. And then, well, in the end, my program would say, well, only this part in the middle, that is what I would trust. And that is what my consensus genome would look like. And that's actually nice, because where I start with millions of particles, then now I would only have like a few thousand of these parts of consensus genome. I'm just left with the problem that I still don't know what kind of genomes I'm actually building, because these are not classified yet. So I use another program that starts to match where these particles belong to. Most of the time, what I find is human or bacterial. And then actually, when I filter out the viruses, I only get 50 virus particles maximum per sample. And that's actually nice, because you can see I have blonde hair, and 50 is a number that I can actually manually interpret even. And from these 50 or up to 50 viral particles, if I would check how much of these particles our Coronaviridae family reads, then it would be 10 to 60%. The other viruses I found were part of my positive controls. I actually spike in, so it's also not crazy that I'm also building back the genomes of my positive controls. But that I find the Coronaviridae is again evidence that there's a Coronaviridae virus in my samples. And actually the programs also give me the percentage, how much they match the old coronaviruses. And now I found that it would match for 72 to 92%. And that would strongly indicate that we have something that really resembles corona, but it's not the same as what we usually, or that we already know. Because if I would sequence a coronavirus that we know and put it next to the genome of a coronavirus that we already have, then you would have like a more than a 99% match. So the fact that we have a lot of these particles that resemble a coronavirus, and then these high percentages that it looks like a coronavirus, that would really be an indication that we are maybe discovering a novel coronavirus, and that also a clinician should maybe isolate this patient as quickly as possible. And we are actually implementing this in our analysis. And then we hope that if this patient pops up at the LUMC, that at least by isolating this patient as quickly as possible, that we can prevent the next pandemic. And I really think that also all large labs around the world, especially in the regions where these viruses emerge, should do more with this metagenomic sequencing and these modern techniques, and also do more on data analysis. So we can also keep the surveillance and the vigilance up, to prevent another pandemic from ever happening again.
Yeah, fantastic. Thank you for breaking it down. I think the Legos are a very good way to explain how next generation sequencing works. And I guess, the nice benefit also of this novel characterization is found in LUMC. Janssen is a stone throw away, so I'm sure they'd be very happy to collaborate very quickly to develop a new novel vaccine, as they're currently doing. I guess, where to start with the questions? Initially for me, so I guess I knew a bit about bacterial metagenomics because that was previously done to characterize beer. But that's because a lot of the strains are already known. So for viral metagenomics, and we're trying to look to prevent the next pandemic, it's going to be a lot of unknowns. Or is that something that still needs to be built, or is that something that is the more data that's being put into the bioinformatic tools, that it can be sort of more mapping it and sort of predict how a specific new strain of pandemics would be developed? Or is it still something that has to come up? Yeah, sorry.
Well, I think we cannot really predict how the next new virus looks like, if that's what you mean. I think there are methods, like the one we also use in the LUMC, that you could at least predict it's a new or a novel virus within a family that we already know. I only know of one software tool that actually, without knowing what kind of family it belongs to, it starts to see like, normally genes of a virus are built up like this. And there's one software tool called VGAS that's actually tries to build genes from scratch without knowing whatever family it belongs to. But it's not tested a lot. I think that would be the next step, actually. but yeah, I hope that a lot of people are aware now that viruses are still a threat. Normally, people think, oh, Ebola, it's just the west coast of Africa, and only some healthcare workers that go there are at risk, and all the other viruses we get vaccinated and it's not so much of a problem. But still, these emerging viruses are a problem. So I think still more research is needed in this field.
So,
Yes.
sorry, go on, Laura.
We got some questions from the audience.
Yeah.
So Natasha asks Xavier if the proteases that you talked about can be used as a target for antiviral development, and if you are looking into that in your research.
Well, definitely. That's one of the main targets. It's one of the main viral proteins that's being targeted for antiviral development. So, because if you block these proteases from cleaving the big polyprotein, then the virus will be unable to replicate and to give progeny. I'm not myself looking into small molecules to block the proteases. I'm rather trying to identify host substrates of these proteases and understand what is the effect of such cleavages. And then maybe this could lead at a longer stage to host-directed therapies rather than virus-directed therapies.
Yeah. Okay.
We have another question. This one is from Guillermo, so I'll pop it up again. Yeah, so I guess this is more as going for the testing that we've discussed. So preliminary hunch is needed. Is there no generic test, or can it be developed for specific viruses?
For all the regular qPCRs, you need to have primers for a specific virus, and you have to put it in for the virus you expect. The metagenomic testing, that is really the test where we look at everything, and also the bacteria pop up. But we are still researching if that's really as reliable as the viral results. But that's more of a generic test.
Okay. I don't know if we still have time maybe for a last question. One person commented that the virus mutates, and if you maybe can explain a little bit how that happens. And I was also wondering if those mutations can somehow inhibit the PCR reaction. So because the virus is continuously changing, and you now have primers for a specific sequence. So if that sequence changes, if the virus changes, can you still identify it on the samples, or is that a problem at all, or is it still easy to identify the virus?
Well, the primers are actually designed in a way that they are in a quite conservative region of the virus, so that it would not mutate quickly. And I think also if there would be a mismatch, then there are still enough DNA matches that remain there to at least amplify something. So it would not all of a sudden, I don't believe so all of a sudden a patient would remain negative, even though it's still there, but with just 1 point mutation, for instance. And we're also sequencing enough to see what's actually happening with all these patients, how are the mutations starting, also to monitor a bit, to prevent also this from happening.
Yeah. Okay. I guess we have a jokester that's watching us. Here's I guess, last question. Hopefully we can answer this quickly. More on the lines of, there's a lot of conspiracy theories. What are your thoughts of the one that's being created in the Chinese laboratory?
I would doubt that. I mean, there's already been several outbreaks of many other viruses during history. And the fact that we are getting also closer because exploiting natural areas that we've never been in touch to also brings animals closer to us, and at that moment the possibilities for a virus to jump from animals to humans is bigger. And yeah, and actually most of the scientific community has proved that that the virus most probably does not come from a Chinese laboratory. At the end, it very much resembles to viruses of bats, and yeah, the possibilities of this happening are very high. In in 2 decades, it has happened 3 times that the coronavirus has jumped from an animal to humans. So I would expect this to be a common phenomenon. Well, hopefully not that common, but something that can happen and that we have to be aware. And it's not that easy to create a virus in the lab.
All right. Thank you.
All right. Thank you for joining tonight. It was really nice to hear more about how viruses infect us, and yeah, how can scientists diagnose the virus from human samples?
Yeah, and I think there's a lot of news that you hear. It's also nice to hear directly from the people doing the frontline research and having to deal with the people that sort of not like wearing masks. So yeah, thank you.
Well, thank you for the invite.
Yeah, thank you.
All right. Thank you to all our listeners. We'll be back in the near future, so stay in tune. And have a good night, all. Bye.
Bye.
Bye-bye.
Bye.
Cheers.